Hello, my dear students, and welcome back. I know your exams are just around the corner, so today we are going to tackle a super important topic, biomolecules. Just imagine that our bodies are made of trillions of tiny little cities, and we call these cities cells. So what are biomolecules? Well, they are the essential materials that build and run absolutely everything in those cities. Come on, let's dive in. To make this chapter bilkul simple for you, we've broken it down into these 6 easy parts. We're going to go through them one by one, step by step, to make sure you are totally ready for your exam. Cello, let's begin. So, just like I was saying, picture each of your cells as this tiny, bustling city. You know, a city needs buildings, it needs roads, it needs power plants, and of course, communication systems. The materials that we use to build and run this entire city, well, that's what we call biomolecules. And here they are, the 4 heroes of our chapter: carbohydrates, lipids, proteins, and nucleic acids. Please remember these 4 names. They are the absolute key to understanding how our bodies work. Okay, let's start with our first building material. Just like any city needs electricity to run, our cells need a constant supply of energy. And where does this energy come from? It comes mainly from our very first biomolecule, carbohydrates. So the main thing to remember here is that carbohydrates are our body's primary fuel. It's just like petrol for a car, right? They provide the energy that keeps every single one of our cells going. Without them, everything would just stop. Now, carbohydrates are classified into 3 simple groups, and it's all based on their size. Think of it like this. First, you have one single brick. That's a monosaccharide. Then you join a few bricks together. That becomes an oligosaccharide. And when you join hundreds and hundreds of bricks to make a long wall, that's a polysaccharide. Simple, isn't it? And here you can see the structure of those basic sugar bricks, the monosaccharides. The 2 most common examples are glucose and fructose. All the bigger carbohydrates are simply made by joining these little units together again and again. And when we join many, many glucose units, we get these huge polysaccharides. Just look at this table. They have very different jobs. Starch, that's like the main energy warehouse for plants. Glycogen is the backup power generator for animals, like us. And cellulose, it builds the strong, tough walls for plant cells. Okay, so we've got our energy source for the city. Now what about the city's walls and its long-term savings plan? That's where lipids come in. In our cell city, lipids have 2 super important jobs. First, they form the boundaries of the city. And second, they are the city's long-term energy savings account. Now, I'm sure you always hear about saturated and unsaturated fats. The difference is actually very simple better. Saturated fats have these nice, straight chains with no double bonds, so they can pack together very tightly. But unsaturated fats, they have double bonds that create little kinks or bends in the chain, so they just can't pack together so neatly. And this molecule right here, a triacylglycerol, this is how our body stores fat. When we have extra energy, our body is smart. It doesn't waste it. It converts it into these molecules and saves it for later. It's literally our body's energy savings account, for a rainy day. Now, a very special type of lipid, the phospholipid, has an absolutely crucial job. It's what makes up the cell membrane. It forms the outer wall of every single cell in our body, keeping the inside of our cell city separate from the outside world. So we have fuel from carbohydrates and walls from lipids. But who does all the work in the city? Let's move on to proteins. If carbohydrates are the fuel and lipids are the walls, then proteins are, well, they're pretty much everything else. They are the workers, the machines, the messengers. They do all the important jobs in our cell city. They are truly the master builders. So what are these amazing protein machines made of? They're built from these small units called amino acids. You can see the basic structure here. There are 20 different types of amino acids, and the only thing that changes between them is this R group. You can think of them like 20 different colored Lego blocks. So how do these amino acid Lego blocks actually join together? Well, they are linked by a very strong special bond called a peptide bond. You can see it forming right here. This strong bond is what creates the long chain, and that's the very first step in making a protein. Okay, Beta, now listen very, very carefully. This is a 5-star important topic for your exams, so pay close attention. A protein isn't just a long chain. It has to fold up into a very specific 3D shape to do its job. There are 4 levels to this folding. First, primary is just the sequence of amino acids. Second is secondary, the first local folding. Third is tertiary, which is the final, complete 3D shape of one chain. And finally, quaternary is when multiple folded chains all come together to work as a team. So let's look at the two main types of that secondary structure. The alpha helix, which you see here, it looks a bit like a spring or a coil. And the other common shape is the beta-pleated sheet, which kind of looks like a folded sheet of paper. These are the very first folds that a protein chain makes. All right, we've got our fuel, our walls, and our workers. But where do the instructions to build all of this even come from? We now come to our final and perhaps the most important biomolecule of all, nucleic acids. If our cell is a city, then the nucleic acids are the master blueprint, the instruction manual, the city's entire codebook that has the plans for everything.
This slide right here is another one of those favorite exam questions. What are the differences between DNA and RNA? It's so simple. Look here. DNA has deoxyribose sugar, while RNA has ribose. DNA uses the base thymine, or T, while RNA uses uracil, or U. And finally, DNA is a double helix, like a twisted ladder, while RNA is just a single strand. Just remember these 3 points.
The DNA double helix is held together by a very strict rule. It's called the base pairing rule. Adenine, or A, always pairs with thymine, or T, and guanine, G, always pairs with cytosine, C. This simple ATGC rule, that is the foundation of the entire genetic code. It's the secret code of life. Wow, you have all done so well. We've covered all 4 of the major biomolecules. Now, before we finish, let's just put that knowledge to the test to really make it stick for the exam. Are you ready for a couple of quick questions? Okay, first question for you. Think back to our carbohydrate section. Which big polysaccharide is the main way that we animals store our energy? Is it starch? Is it cellulose? Or is it something else? Take a second to think about it. Did you get it?
The answer is glycogen. Remember, starch is for plants, but glycogen is for animals. Very well done if you got that right.
Okay, just one more question, this time about proteins. We have all those amino acid Lego blocks. What is that special name for the bond that links them all together in a long chain?
And the answer is a peptide bond. Fantastic! You all are paying such good attention today. I am very, very proud of you.
See? You know this chapter so well now. You are absolutely ready to ace this topic in your exam. Just keep revising these key points, my dear students. Don't stress. Don't worry at all. You have worked hard, and you have totally got this. All the very best.